Hello. Today, for my exam 2 mini video, I'm going to be looking at the genetic mech for dragons. So I chose the dragon because it would be very interesting to look at how the genetics of the dragon work, and also it's a very unique character in Game of Thrones and one of my personal favorite creatures. So first, we're going to look at 3 different traits within a dragon on this one chromosome. We're going to look at scale color, which can either be green or blue. We're going to look at tail shape, which can be spiky or flat, or fire breath, which can be fire or ice. Here I've indicated the dominant genotype being a capital letter here. So green is dominant to blue, big S spiky would be dominant to flat, and big F fire is dominant to ice. So over here we have our father genotype, which is homozygous in every category. We have big G, which is green, little s, which is a flat tail, like we have here, and then we have fire breathing. And over here we have a mother who is little g blue, big S spiky tail, and little f ice, which are all homozygous here. We also have region 1 being 13 centimorgans and region 2 being 15 centimorgans, and same thing over here. So how we get the so we first cross the mother and the father, which end up in a genotype of all heterozygous traits. So over here we have mother on the top and father on the bottom, with region 1 and region 2 being again 13 centimorgans and 15 centimorgans. And next we're going to look at the probabilities of each gamete. So in this table here, I have each specific gamete that is listed with a no crossover, meaning it goes straight across with no crossover over the regions. Double crossover, which means crossing over both of the regions. Single crossover region 1, which means it crosses over only in region 1. And a single crossover 2, which means it only crosses over in region 2. Up here, I also have the coefficient of coincidence at 0.9, and the probability is listed here for every single possible gamete type. So how do we calculate these? So for a double crossover event for one gamete, the calculation goes as follows. So first you multiply region 1, which is 13 centimorgans, times region 2, which is 15, times the coefficient of coincidence. And then you get that number, which then you multiply by half, because there are 2 different gametes that could possibly show up. So for one gamete specifically, which is GSF, it would be that. So for single crossover 1, you kind of do the same, where you have region 1 right here. You subtract the double crossover event, which you calculate in the last slide, which would be this number. And then you would multiply it by a half, or subtract it from the region 1, which then multiply it by a half to get this number, which is then only for one one specific genotype and one specific gamete. Over here, I have my references, which I use, including images and information.